Welcome everyone. Let's begin with the fundamental question: what is genetics? Genetics is the branch of biology that deals with heredity and variation in all living organisms. Let me break that down. Heredity is the passing on of traits from parents to offspring. This is why children resemble their parents, why certain diseases run in families, and why you might have your grandmother's eye color. Heredity is what makes us similar to our relatives. Variation refers to the differences between individuals. Why do siblings from the same parents look different? Why do some people have blue eyes while others have brown? Why does one person develop a genetic disorder while another one doesn't? Variation is why we are all unique. Genetics seeks to answer all of these questions. And notice the definition says in all living organisms, from bacteria to plant to animals to humans. The fundamental principles of genetics are universal. In summary, genetics is the science of heredity and variation. This is the foundation for everything we will study in medical genetics. Now that we understand what is genetics, let me look at how this broad field is organized into subfields. Genetics is a vast discipline and it is divided based on the type of organism being studied. The slide shows 4 major subfields: human genetics, animal genetics, plant genetics, and medical genetics. Let me explain each one. First, human genetics. This is the study of heredity and variation, specifically in humans. Human genetics study how traits, both normal traits like eye color and blood types, and disease traits like phenylketonuria or cystic fibrosis, are passed on through human families. Human genetics uses tools like pedigree analysis, twin studies, and molecular techniques to understand human inheritance. This subfield is the foundation for understanding all human traits and diseases. Second item is animal genetics. This subfield focuses on heredity and variation in non-human animals. Animal genetics is used in many practical applications, such as breeding better livestock, preserving endangered species, studying animal models for human diseases, and even understanding the genetics of our pets. Third item is plant genetics. Plant genetics study heredity and variation in plants. This subfield has enormous practical importance. Plant genetics is used to develop crops with higher yields, better resistant to pests and diseases, tolerance to drought and heat, and improved nutritional content. The Green Revolution, which saved millions from starvation, was built on advances in plant genetics. Today, plant genetics includes the development of genetically modified crops that can feed our growing global population. Fourth item, and the most relevant to many of you, is medical genetics. Medical genetics apply genetics principles to understand, diagnose, treat, and manage genetic disorders in humans. This is the clinical application of human genetics. Medical geneticists work with patients and families affected by conditions like Down syndrome, cystic fibrosis, Huntington disease, and cancer. Medical genetics includes genetic counseling, prenatal diagnosis, newborn screening, and the emerging field of gene therapy. Now, you might notice that these subfields overlap. Human genetics and medical genetics are closely connected. Animal genetics and plant genetics often use similar molecular tools. The boundaries between subfields or field techniques developed in plant genetics might later to be applied in human medicine. Now let's focus specifically on medical genetics, the subfield most relevant to your studies and future careers. What is the medical genetics? Medical genetics is a branch of genetics that applies genetic principles to understand, diagnose, treat, and manage genetic disorder in humans. It bridges the gap between basic genetic science and clinical medicine. What does a medical geneticist do? Medical geneticists evaluate patients with suspected genetic condition, order and interpret genetic tests, make diagnoses, and manage treatment. They work with conditions such as phenylketonuria, Down syndrome, cystic fibrosis, Huntington disease, cancer genetics, and many others. What are the key components of medical genetics? Medical genetics include several specialties, such as clinical genetics, with patient evaluation and diagnosis, or cytogenetics, a study of chromosomes and chromosomal disorder, and molecular genetics, which deal with DNA analysis to detect gene mutations, biochemical genetics, which deals with analysis of metabolites to diagnose metabolic disorders, genomics, whole genome approaches to understand complex diseases. Why is medical genetics important? Medical genetics allows us to provide accurate diagnosis for patients with genetic disorders, offer prenatal diagnosis to at-risk couples, perform newborn screening to detect treatable conditions early, provide genetic counseling to help families understand recurrence risk, develop targeted treatment based on genetic finding. Let's discuss the three fundamental principles of genetics that Mendel deduced from his experiments with pea plants. First, segregation. Mendel observed that each individual has two copies of each factor, what we now call genes, and these two copies separate or segregate during gamete formation. Each parent passes only one copy to each offspring. This explains why children inherited one allele from their mother and one from their father. Second, dominance. Mendel noticed that when an individual has two different factors or two different alleles, one may mask the expression of the other. The allele that is expressed is called dominant, and the allele that is masked is called recessive. Third, independent assortment. Mendel found that factors for different traits are inherited independently of each other. The gene for seed color doesn't influence the inheritance of the gene for seed shape. This principle holds true for genes located on different chromosomes. But we now know that genes on the same chromosome may be linked. These three principles, segregation, dominance, and independent assortment, form the foundation of classical genetics and remain essential for understanding inheritance patterns in all organisms, including humans. In this slide, we're talking about conceptual history of medical genetics. In 1901, the dominant inheritance of brachydactyly was discovered. In 1902, the inborn error metabolism of disease was discovered. In 1918, described anticipation. I'm talking about it later. In 1931, cytoplasmic inheritance of mitochondrial DNA was discovered. In 1937, linkage of color blindness and hemophilia was determined. In 1955, they found that human diploid chromosome number is 46. In 1970, the scientists did amniocentesis for chromosomal disorders. In 1970 also, they did screening for Tay-Sachs disease, which is common in Jewish people. In 1976, the human globin gene was cloned. And also in 1985, the technique called PCR, or polymerase chain reaction, was identified. In 1986, the gene for the disease of Duchenne muscular dystrophy was determined. In 1986, also the gene for cystic fibrosis was identified. In 1987, the scientists did predictive genetic testing for Huntington disease. In 1998, the scientists decided to sequence entire human genome. In 1991, medical genetics became an specialty in American Board of Medical Science, and also in the same time the first draft of human genome was identified, and in 2001 the human genome sequence completed. The genetics back to Mendel's law. In 1860, Mendel worked on peas. They found three laws that became these three laws as a basic of classic Mendelian genetics. He found some traits are inherited through one generation to another generation. In 1870, the scientist Friedrich Micher described nucleotide acid. In 1909, the word gene, coined by a Danish botanist, William Johansen, and in 1910, Morgan work on fruit flies demonstrates that genes lie on chromosomes. In 1940s, McClintock described mobile genetic elements in maize. In 1944, Avery shows in bacteria that nucleic acids are the transforming principle. In 1953, Watson and Crick published the double helix model for DNA chemical structure. In 1977, Sharp and Roberts find the protein coding genes are carried in segments. And in 2001, the initial results from the Human Genome Project published in Journal of Nature and Science. Medical genetics since 1950s to the present. In 1953, Watson and Crick described the DNA structure as a double helix. And also, in 2003, the Human Genome Project, they finished. And after 2003, the future of medical genetics focused on diagnostic and treatment. First time in 1970, they used ultrasound and amniocentesis for prenatal diagnosis of genetic disease. After that, they focused on complex genetic disease such as non-Mendelian genetics, genomic imprinting, triple nucleotide repeats disease, mitochondrial inheritance, and since 1990s, the scientists focused on neuropsychiatric disorders, diabetes, and cardiovascular disease. And also after that, the scientists focuses more on interaction of genes with environmental triggers. Okay, when we are talking about the genetics, we have to know some basic things such as what is gene? Gene is a basic unit of genetic information. Genes determine the inherited characters. And what is genome? Genome is the collection of genetic information. And the gene is stored in a unit called chromosome. And the gene also contains DNA. And what is DNA? DNA is a nucleic acid that contains the genetic instruction specifying the biological development of all cellular forms of life. Let's clarify two essential terms for understanding chromosome structure: locus and allele. First, locus. The word locus is Latin for place or location. In genetics, a locus refers to the specific fixed position of a gene on a chromosome. Think of it like an address. Just as every house has a unique street address, every gene has a unique locus on a specific chromosome. For example, the PAH gene is located at locus 12q, meaning on chromosome 12 on the long arm or Q. Now allele. An allele is one variant form of a gene at a particular locus. Since humans are diploid, we have 2 copies of each chromosome and therefore 2 copies of each locus. These 2 copies may most human cells contain 46 chromosomes. As you can see here, from these 46 chromosomes, 22 pairs of chromosomes named autosomes, which is common between male and female, and the differences between chromosomes between male and female is sex chromosome. In male, they have two sex chromosome X and Y, and in female, they have two X chromosomes. Let's define four essential terms in genetics: phenotype, genotype, mutation, and polymorphism. First, phenotype. This is the physical description of a character in an individual organism. It's what you can observe: eye color, height, blood type. Phenotype results from both genotype and environmental influences. Second, genotype. This is the genetic constitution of the organism, the specific combination of alleles an individual carries. Now, the next two terms both refer to changes in genetic material, but with an important distinction. Mutation is a change in a genetic material that is usually rare and pathological, meaning it causes diseases. Mutations are typically found in less than 1% of the population. Polymorphism is also a change in a genetic material, but it is usually common and not pathological. Polymorphisms are found in more than 1% of the population and typically do not cause disease. As you can see in this slide, the color of the eyes and also color of the skin, which is a character of inherited, is different in these two individuals. And also the color and structure of hair is also different in these two persons, as the phenotype of them are different. Okay, what we have learned from our genome so far, there are a relatively small number of human genes, less than 30,000, but they have a complex architecture that we are only beginning to understand and appreciate. We know where 85% of genes are in a sequence. We don't know where the other 15% are because we haven't seen them on, and maybe they are only expressed during fetal development. We only know what about 20% of our genes do so far. So it is relatively easy to locate genes in a genome, but it is hard to figure out what they do. In human beings, about 99.9% of nucleic acids or DNA are the same, with remaining about 0.1% makes a person unique. These different the genotype cause a person look specific, and also a disease for he or she may be developed during the mutation in this 0.1% of nucleic acid. These variations can be harmless, just maybe change in phenotype, or may be harmful, cause the disease such as diabetes, cancer, heart disease, Huntington disease, and hemophilia. And this variation can be latent. Variations found in coding and regulatory region are not harmful on their own, and the change in each gene only become apparent under certain conditions, such as susceptibility to heart attack. And what is the importance of genetics to medicine? We know that about 50% of first trimester abortion are due to chromosomal abnormalities, and also about 2 or 3% of newborn they have got congenital malformation, and 2% infants are born with single gene disorder, and also more than half of childhood blindness, deafness, and mental retardation are due to um, genetic disorders. Another importance of genetics to medicine is changing focus of medicine on primary care physicians in front of specialists, and also prevention in front of treatment, and also genetic causation of both rare and common diseases, and also there are another importance about human genome project, and also due to information we get through this human genome project, nowadays we can design drugs. Genetics also can link between basic research and clinical observation. Okay, the categories of genetic disease. Some of the disease are multifactorial disease, which is a polygenic condition. It means several genes involved in this type of genetic disease. And some of this multifactorial disease is caused because of interaction of genetic and also environmental factors. Another type of genetic disease is either because of whole chromosome loss or deletion of whole chromosome, or maybe partial chromosome loss or insertion. And another type of disease is about the single gene disorder. The single gene disorder inherited, such as Mendel's rule. Mendel's rule says the disease may be autosomal dominant, autosomal recessive, or X-linked. Autosomal recessive, it means for the disease who is inherited as autosomal recessive, both parents should be carrier of the disease, and in 25% of offspring they show the disease. An X-linked disease is just the boys show the disease because they have got just one X inherited from his mother. I've summarized all of the genetic disease in this slide. Genetic disease is categorized by three parts. One type of genetic disease is chromosomal disease. One part is because of single gene disorder, and the other one is multifactorial. If it is chromosomal disease, maybe the changes happen in a chromosome, either is numerical change or structural change, or some micro deletions. When happen on a chromosome, the disease will appear. And also, if it is single gene disorder, the inheritance can be autosomal dominant, autosomal recessive, X-linked, and maybe mitochondrial disease, which is inherited just from mother. And another part of disease is multifactorial. If you look at this slide, it shows the contribution of genetic and environmental factors to human disease. Some type of disease which is multifactorial, they have or they're susceptible to have genetic background, and then when they face with the specific environmental factors, the disease will appear, such as diabetes or peptic ulcer or even tuberculosis. And some part of the disease is specifically the gene is the deletion or mutation in a gene can cause that type of disease, such as phenylketonuria, galactosemia, or osteogenesis imperfecta, hemophilia, and Duchenne muscular dystrophy. This type of disease is rare, and then the cause of this disease is a one or two gene. It means it is unifactorial and high recurrence rate. But in multifactorial disease, these type of disease are common, and low recurrence rate has happened in this type of disease. Okay, I'm talking about some fundamental terminology in the field of the genetics. Let's define heredity. Heredity refers to the passing on of traits from parents to offspring. This is the phenomenon we have all observed. Why children resemble to their parents, why certain diseases run in families. This concept has been recognized for thousands of years, long before we understood the molecular basis. Genetics, in contrast, is a scientific study of heredity and its variations. While heredity describes the observation, genetics is a science that seeks to explain the mechanisms. Genetics asks how are traits transmitted? Why do variations occur? What molecules are responsible? Now, medical genetics takes this one step further. Medical genetics is the application of genetic principles to understand, treat, and manage genetic disorders. This is what many of you will practice. It bridges basic genetic science with clinical medicine. For example, when we study PAH gene mutations causing phenylketonuria, we are doing genetics. When we then use that knowledge to offer prenatal diagnosis or design treatment strategies, we are practicing medical genetics. Finally, who are the professionals in this field? Medical geneticists are physicians with specialized training in genetics. They evaluate patients, order genetic tests, interpret results, and manage treatment. Genetic counselors are master degrees level trained healthcare professionals who help patients and families understand genetic and conditions, the risk of inheritance, and options available. They provide psychological support and facilitate informed decision making. So, in summary, I have to tell you, heredity is the phenomenon, genetics is the science. Finally, let's discuss one of the oldest and most fundamental tools in medical genetics, the pedigree. What is pedigree? A pedigree is a schematic diagram that indicates the structure of a family and the transmission of a trait or disease across generation. It is a visual representation of family relationship and the presence or absence of a particular phenotype. Standard, uh, Standardized symbols are used. A square represent male, circles represent female, field symbols indicate affected individuals, half-field symbols indicate carriers, diamond indicates individuals of unspecified sex. Vertical line from a mating line to a symbol indicate offspring. Roman numeral indicate generation. Arabic numerals indicate individuals within a generation. And what is a pedigree analysis? Pedigree analysis is a process of analyzing traits within the families to reveal fundamental patterns of inheritance. By constructing a pedigree and observing how a disorder is transmitted, a geneticist can determine the most likely inheritance pattern. What pattern can pedigree analysis reveal? Autosomal dominant appears in every generation, affected both sexes equally. Affected parent can pass about 50% of children. Autosomal recessive may skip generations, affects both sexes equally. Unaffected parents can have affected child. X-linked dominant affect both sexes but more females. No male to male transmissions. X-linked recessive primarily affects males, carrier female are carrier, no male to male transmission. Y-linked is only male affected, passed from father to all sons. Mitochondrial pattern is transmitted from mother to all children.